Good evening. This is Pangnuri Ajay Kumar, and the topic is gastrointestinal physiology. Gastrointestinal physiology is a fascinating and complex field that encompasses the study of the structure, function, and regulation of digestive system. The system plays a vital role in our survival, breaking down food into nutrients that our body can absorb and utilize. This presentation will delve into the intricacies of gastrointestinal physiology, exploring its diverse components and their intricate interplay. From the anatomical structures of gastrointestinal tract to the complex mechanisms of digestion, absorption, and regulation, we will embark on the journey through the remarkable world of digestive system. Structure and innervation of gastrointestinal tract. Layers of the gastrointestinal tract. The GI tract and the GI tract wall comprises four distinct layers: the mucosa, submucosa, muscularis propria, and serosa. The mucosa is the innermost layer responsible for absorption and secretion, while the submucosa houses blood vessels, lymphatics, and nerves. The muscularis propria facilitates peristalsis, propelling food through the tract, and the serosa is the outermost layer protecting the GI tract. Enteric nervous system. The enteric nervous system is a complex network of neurons associated or neurons located with the walls of the GI tract. This is also called as the brain in the gut, controls various aspects of digestion, including motility, secretion, and absorption, dependent of central central nervous system. The ENS operates through two plexures, the, the myenteric plexus regulating muscle contraction and submucosal plexus managing secretion and blood flow. Autonomic nervous system influence. The autonomic nervous system, specifically the sympathetic and parasympathetic branches, also influences GI tract function. The sympathetic nervous system responsible for the flight or fight response inhibits GI activity, while the parasympathetic nervous system, promoting rest and digest, stimulates GI processes. This interplay ensures appropriate digestion and nutrient processing based on the body's overall needs. Regulatory substances in the GI tract. Gastrointestinal hormones. Gastrointestinal hormones are secreted by endocrine cells within the GI tract and act on the target cells to regulate various processes. Examples include gastrin stimulating gastric acids secretion, cholecystokinin promoting bile release and pancreatic enzyme secretion, and secretin triggering bicarbonate release from the pancreas. Neurotransmitters like acetylcholine, norepinephrine are released by neurons within the enteric and autonomous nervous systems. Acetylcholine stimulates GI motility and secretion, while norepinephrine inhibits these functions. These neurotransmitters play a crucial role in coordinating digestive processes within the body's overall needs. Cytokines. Cytokines are the small protein molecules produced by the immune cells within the GI tract. They contribute to inflammatory response and influence various aspects of digestion, including motility, secretion, and permeability. Cytokines also plays a major role in gut microbiota interactions, contributing to overall health of digestive system. Local factors. In addition to hormones, neurotransmitters, and cytokine, local factors like pH, osmolarity, and nutrients also influence gastrointestinal function. These factors trigger local responses, ensuring appropriate digestion and absorption based on the specific content of gut lumen. Gastrointestinal motility. Peristalsis is the coordinate rhythmic contraction of muscularis propria that propels food through the GI tract. The circular muscles contract behind the food bolus, while the longitudinal muscles relax, pushing the bolus forward. This wave-like motion ensures the efficient movement of food from the esophagus to the rectum. Segmentation. Segmentation is a type of motility primarily observed in the small intestine. It involves in the rhythmic contraction and relaxation of circular muscles, mixing the intestinal contents and promoting the optimal contraction between the chyme and the intestinal walls for nutrient absorption. Segmentation does not propel the food forward, but rather mixes it from for better digestion. Factors influencing motility. Gastrointestinal motility is influenced by various factors, including the enteric nervous system, the autonomic nervous system, hormones, and the local factors like and nu- uh, pH and nutrient content. These factors ensures appropriate motility patterns based on the digestive needs of the individual and the type of food being processed. Gastrointestinal secretion. Starting with the saliva, the source of saliva is salivary gland, and the components of saliva includes water, electrolytes, enzymes like amylase and lipase, which helps in the lubrication, digestion of carbohydrates, and a mild antibacterial action. Gastric juice, it is secreted from stomach. Components include pepsin, mucus, and intrinsic factor. It helps in the acidification, protein digestion, protein of protect stomach lining, and vitamin B12 absorption. Pancreatic juice, secreted by pancreas. It contains enzymes, proteases, amylases, lipases, nucleases, and bicarbonates. It helps in the digestion of proteins, carbohydrates, fats, and nucleic acids. It also neutralizes the chyme. Bile, it is secreted by liver and stored in the gallbladder. Bile salts, bilirubin, and cholesterol. It helps in the emulsification of fats, excretion of waste products. Digestion and absorption. Carbohydration. Carbohydration begins in the mouth with salivary amylase breaking down the starch into smaller sugars in the small intestine. Pancreatic amylase continues the process further, breaking down the carbohydrates into monosaccharides like glucose, fructose, and galactose, which are then absorbed into the bloodstream. Protein digestion. Protein digestion commences in the stomach with pepsin, which breaks down the large proteins into smaller polypeptides in the small intestine. Pancreatic proteases like trypsin and chymotrypsin further break down polypeptides into amino acids, which are absorbed in the bloodstream. Fat digestion. Fat digestion begins in the small intestine with the help of bile salts secreted by the liver. Bile salts emulsify fat, breaking them down into smaller droplets, increasing their surface area for digestion by pancreatic lipase. The resulting fatty acids and the glycerol are then absorbed into the bloodstream. Coming to the liver physiology, bile production. The liver produces bile, a fluid essential for the digestion of fats. Bile salts, the main components of bile is to emulsify fats, breaking them down into smaller droplets, increasing their surface area for digestion by pancreatic lipase. Second function is detoxification. The liver acts as a detoxifying organ, removing the harmful substances from the bloodstream. It metabolizes drugs, alcohol, toxins, and waste products, converting them into less harmful forms that can be excreted. This function is essential for maintaining overall health. Nutrient metabolism. The liver plays a crucial role in nutrient metabolism, converting the carbohydrates into glucose, synthesizing proteins and storing and releasing fats. It also regulates blood glucose levels, ensuring a steady supply of energy for the body. Other functions of liver include vital functions like production of clotting factors, albumin, and other protein essential for blood coagulation and fluid balance. It also stores vitamins and minerals contributing to the overall health and well-being. Gut microbiome and its importance. It has several functions, starting with the immune function enhancement. The gut microbiome plays a crucial role in shaping the immune system, influencing its development and function. It helps to train the immune system to recognize and respond to pathogens while tolerating harmless commensal bacteria. Normal digestion, nutrient digestion, and absorption. Gut microbiota contribute to nutrient digestion and absorption by producing enzymes that breaks complex carbohydrates like fibers, which the human digestive system cannot digest on its own. This process enhances the utilization of nutrients from the food. Mental health regulation. Emerging evidence suggests a connection between the gut microbiome and the mental health. The microbiota then can produce neurotransmitters like serotonin and GABA, which influence mood, cognition, and behavior. Disturbances in microbiome composition may contribute to mental health disorders. Protection against pathogens. The microbiota forms a protective barrier against harmful pathogens, competing for resources and producing antimicrobial substances that inhibits the growth of pathogenic bacteria. This barrier helps to maintain the health of digestive system. Common gastrointestinal disorders. First one is irritable bowel syndrome. IBS is the most common disorder characterized by abdominal pain, bloating, gas, diarrhea, and constipation. It is believed to be caused by combination of factors including genetics, stress, and changes in the gut microbiota. Inflammatory bowel disease or IBD encompasses Crohn's disease and ulcerative colitis. Chronic inflammatory conditions affecting the digestive tract. IBD is characterized by inflammation, ulcers, and abdominal pain, and its exact causes are unknown, but unlikely involve genetic predisposition and environmental factors. GERD or gastrointestinal reflux disease. GERD occurs when the stomach acid flows back up into the esophagus, causing heartburn, chest pain, and difficulty in swallowing. It is often caused by weakened lower esophageal sphincter, which normally prevents acid reflux. Peptic ulcer disease. Peptic ulcer disease refers to sores that develops in the lining of stomach duodenum. It is mainly caused by Helicobacter pylori or overusage of non-steroidal anti-inflammatory drug or stress. Advances in the gastrophysiology research. Microfluidic devices are miniature labs that mimics the microenvironment of the gastrointestinal tract. These devices enable resources to study the interactions between cells, enzymes, and other molecules in a controlled and physiologically relevant environment. Organ on chip models. Organ on a chip models are three-dimensional cultures of human cells that recreates functions and structures of specific organs, including the gut. These models provide a more realistic platform for studying drug efficacy, toxicity, and disease mechanism. Personalized medicine approaches. Personalized medicine approaches aim to tailor treatments to an individual patient's genetic makeup, microbiome composition, and lifestyle factors. This approach holds a greater promise for improving the diagnosis and treatment of gastrointestinal disorders. Thank you.